Hi, this is Angie Drakulich, Senior Managing Editor of Pharmaceutical Technology, and today I'm speaking with Dr. Francis Collins, Director of the National Institutes of Health. Dr. Collins is the keynote speaker at the 2011 Biotechnology Industry Organization Convention and Partnering for Global Health Forum, sponsored by Bio and BioVentures for Global Health. Thank you so much, Dr. Collins, for talking with me this morning.
Glad to be able to chat with you, Angie.
Great. Now, since you became director of NIH in 2009, you made it clear that you wanted to make global health a top priority. I was wondering if you could summarize the role you saw then for NIH in global health and whether you feel that major strides have been made thus far.
Yes, in figuring out what were the themes that offered the greatest opportunity for progress in terms of NIH research, I spent a lot of time speaking with experts, seeking advice, and trying to identify areas that would fit as being exceptional opportunities. And global health clearly emerged as one of them and certainly resonated with my own desires to see advances in medical research benefit not only people in high-income countries but also throughout the world. And I do think scientifically the arguments are pretty compelling that this is a unique time for assisting that process through NIH research. After all, we're making discoveries about the nature of pathogens that cause infectious diseases, that suggest new ideas about vaccines and therapeutics. And I think we have increasing abilities also to look at non-communicable diseases in new ways and try to interrupt what otherwise is going to be a cycle where those cause an increasing amount of morbidity and mortality throughout the world. Since I came to this position in August of 2009, so it's a little less than 2 years, I convened almost right away a major gathering of organizations that fund global health research to try to get a better handle on who's doing what and where the gaps might be. And that was quite instructive because there are a lot of players here doing a lot of interesting things, and it's not trivial to figure out exactly where the opportunities lie. There are two projects that have come out of that discussion that are now underway in one instance and about to get underway in another that I think are going to be interesting new NIH investments on a fairly broad scale in sub-Saharan Africa.
Okay.
One of those is what's called the Medical Education Partnership Initiative, which is now funding academic institutions in 12 countries, a total of 30 institutions, to build capacity both for medical education and for research. And we're doing this jointly with PEPFAR. Eric Gooseby and I have agreed that this is a shared area of real importance for that part of the world. And the kickoff of the MEPI program occurred in March in Johannesburg, and it was very exciting to see how all of these investigators coming from institutions all over the continent, many of whom had never really been invited to come to the same meeting together and learn from each other, are really now going to be given a chance with NIH support and support from PEPFAR to build that kind of capacity. I think that's a theme that I'd really like to see highlighted here, which is that the old model where global health research is done by institutions in high-income countries and then sort of offered up to low-income countries has done good things. But in the future, we'd like to see that research capacity built in the countries where the medical needs are so great. And I think we have a chance to do that through programs like MEPI.
Okay.
The other program, which is going to be sponsored jointly by the NIH and the Wellcome Trust, and if you perceive a theme here of partnerships, you're right. This is something called Human Health and Heredity in Africa, or otherwise known as H3 Africa, which is a bold effort to create a network of research activities that will look at environmental and genetic risk factors for both infectious and non-infectious diseases in the continent. Adopting some of the same strategies that have been carried out more recently here in the U.S. and other developed nations, but trying to find out in the cradle of humanity, which is where we all came from, how can we make the most of some of the new technologies that allow us to do genomics and environmental science and understand the causes of illness. This will mean building, again, capacity to do that kind of research, including information networks to share data and to carry out computational analyses. It will include setting up phenotyping capabilities to look at clinical consequences of various exposures and genetic risk factors. And it will mean biorepositories to be able to store the samples that will be collected on what we expect will be thousands of individuals in Africa who will be the research participants in this bold new effort, which will undoubtedly stretch over many years.
Okay, thank you so much. You sort of touched on another topic that I wanted to ask you about, which is, you know, in previous statements you've mentioned that innovation should not be limited to the work that's done for developing nations. And in your opinion, how can we best capture the technology and knowledge that's used for developing nation treatments and apply them to drug development and discovery in every region of the world?
Well, I think of innovation very much as a two-way street, and we shouldn't think of the ways in which new inventions, new creative ideas come forth in the U.S. or in Europe as only going one direction, namely, oh, well, we'll try to figure out how to distribute those to other parts of the world. We can also learn in what's being called reverse innovation about how to adopt new ideas that are being developed in low-income countries. A particularly good example is the use of cell phone technologies for medical purposes, because after all, in many countries in Africa or other parts of the world that are very recently getting communication capabilities, it's cell phones that are driving that. They're not limited by the landline ideas that we all sort of grew up with. Exactly. So, for instance, we have a technology that's being developed and tested in Africa, which is a simple method of assessing whether, in fact, individuals who are being treated for tuberculosis or HIV/AIDS are compliant with the therapy. So you have a pillbox, which essentially is hooked up to the cell phone network, and every time the pillbox gets opened, it sends a signal to the clinic where that patient is being cared for, so that you can tell, was it in fact opened and was it at the appropriate time of day, as a pretty good indication that the medicine is actually being taken as directed, and there hasn't been some confusion about the importance. And, of course, that's crucial for diseases like tuberculosis or HIV/AIDS.
Right, and it sounds like that provides a great way for the doctors to follow through when they're not, you know, close by.
Exactly. And that already has spurred ideas about how we might adopt those same approaches here in the U.S. For people who have, for instance, diabetes or hypertension, where medical compliance, we know, is critical for success, and yet we haven't really had a good way of monitoring that.
Okay. Now, in terms of partnerships, I recently spoke with Dr. Stephen Groft on your team. He heads the NIH Office for Rare Disease Research, and about how that office is coordinating research and clinical trials with other industry and regulatory groups targeting finding treatments for rare diseases. And I was wondering if you found whether countries and other groups are more willing to collaborate in recent years, and if so, what do you think has led to that change?
I certainly do think that collaborative spirit is expanding in a wonderful way, and it's driven in part by scientific opportunities where, for rare diseases, we're discovering the molecular causes at a prodigious rate. There are now some 4,000 rare diseases where we know at a pretty detailed level what the actual molecular problem is that leads to that illness, many of these being genetic diseases caused by mutations in the genome. And so if you want to see that knowledge applied in terms of developing new therapeutics, that is something that is not trivial, and you certainly don't want to waste the opportunity to bring groups together that might be able to do this faster, and you don't want to duplicate efforts and waste resources. So in that regard, the discussions that have been going on in terms of the International Rare Disease Consortium, with particularly strong leadership from the European community, which is this individual, Ruxandra Drakulich, who you may know from this conversation with Steve. In fact, I was with Ruxandra this past Monday in Seattle, where the heads of the international research organizations of the world got together.
Oh, great.
That's a gathering that has been in place now for 10 or 15 years, but it seems particularly vigorous right now So myself and Mark Walport of the Wellcome Trust and Ruxandra from the EC and leaders from China and South Africa and New Zealand and France all sit around the table to talk about ways that we could work together, and rare diseases is a particularly attractive opportunity right now.
And it seems like there's a lot of success coming out of that area as well so far, and with the big targets of having, I believe it's treatments for about 200 diseases
That's the
by
goal.
2020. Right.
200 treatments by 2020. That is bold indeed, because it's already 2011, and we know how long and drawn out that pathway can be.
Right, and it sounds a lot like the Millennium Development Goals and their large targets. Do you think it will be possible to achieve?
I think it's great to set ambitious, even audacious milestones and then challenge people to meet them. This is indeed an audacious goal, 200 new treatments by 2020. We better really start moving fast.
Sounds like a plan. Let me move just back to innovation a little bit. Some other conversations going on in pharmaceutical industry right now is regarding translational research, and NIH is a big supporter of that. I was wondering how NIH is selecting projects to pursue in that area and how far it can take the projects, for example, through proof of concept in animals or phase 1 clinical trials.
So this all ties into the conversation we were just having about rare diseases and the goal of having new therapeutics in a reasonable time. As you know, the current situation is a little scary when it comes to making such promises. Because after all, the average time it takes from identification of a potential drug target to ultimate approval of that therapy is about 14 years, and the failure rate is about 98%. We think the time has come to look at that process the way that an engineer would and see if there are ways that we could optimize some of the steps that currently are slow, expensive, and likely to fail. And that is part of NIH's effort now in putting together a new entity called the National Center for Advancing Translational Sciences. We certainly will do this in a way that is complementary and not competitive with what the private sector would like to do. But we do expect that this kind of initiative may make projects that previously appeared too risky start to look attractive. So some of this is the idea of de-risking projects, which the private sector might otherwise not feel were economically attractive. And certainly for NIH itself, we will, when we see opportunities for therapeutic development, working through the 27 institutes and centers that have a lot of knowledge about these areas, try to move projects forward to the point where they become commercially viable and then license them out as quickly as possible in order to get them over the finish line. Again, the goal here is to try to take advantage of remarkable scientific opportunities that might otherwise lie untouched, but also to recognize the economic realities, which means that companies in general are not going to go after projects that they don't think ultimately will become profitable.
Right. And do you think at some point you'll be able to recruit development partners in industry, such as any big pharma companies?
Oh, absolutely. I think already we're having some pretty interesting conversations with leaders of big companies about ways that we could work together in some of these areas that currently tend to be slow and inefficient. One example, how do we actually determine whether a new potential drug is safe to give to humans? Right now, the way that that is done as a sort of preclinical toxicology depends upon the use of animal testing, which is expensive, slow, and often not very reliable. We have a whole program already underway called Tox 21, as 21 being the 21st century, that is jointly done with the FDA and with EPA, where we're both looking at potential environmental toxins, but also at drugs, to see if there are better ways that are higher throughput, depend upon human cells as opposed to other animals, and it would give a reliable signal about whether a compound is safe or not. And that Tox 21 project is, I think, a good start in the direction of what could be a totally new science of doing preclinical toxicology in the era of having human cells that could be engineered into three-dimensional organoids and that could be a pretty good indicator of whether something is safe or not.
Sounds fascinating. You mentioned funding a little bit in your last answer, and I did want to ask you about a bit of a hot debate topic right now, which has to do with the NIH grant program and small businesses. And from what I understand, the Small Business Innovation Research Program has been authorized by Congress until September 30th of this year, but there's still debates in Congress about how venture capital affects those grants and the qualification of companies. So in your opinion, what is best in that regard for drug discovery and development? Are there certain things you'd like to see happen with that particular bill and the program?
NIH has a long track record of supporting small businesses in a very productive fashion. We can point to some fairly major successes there that have resulted from that kind of starting support for the early phase of a company's development. Take the company Affymetrix, which has been a leader in the whole area of microarrays. That whole company was started on an SBIR grant and is now valued at quite a lot of money. We could point to other examples as well. NIH's perspective is we would like our SBIR dollars to be used for projects that have the greatest chance of ultimately resulting in public benefit, and to have companies excluded on the basis of the amount of venture capital involved in their startup doesn't always make sense. So we are looking forward to seeing that limitation relaxed a bit, because I think it has excluded some companies that might have been really good grantees from NIH to be able to apply. Obviously, that's a topic that's in an ongoing discussion on the Hill.
And another area of leadership and research for NIH is the Genome Project, of course, which is supposed to provide the foundation for personalized medicine. Can you comment on how close we are to truly having personalized medicine, or what you think needs to happen to make it a reality?
Well, I think we're there in some instances, and it isn't one of those things where you don't have it one day and the next day you do. It comes along in various applications bit by bit. If you consider, for instance, a woman who's diagnosed today with breast cancer and has negative lymph nodes at the time of surgery, the question is the surgery she just had, the lumpectomy, and the radiation that will follow, is that sufficient? Is she cured, or does she also need adjuvant chemotherapy? Well, personalized medicine is here because about half of the women in the U.S. who are in that situation this year will have their breast cancer cells analyzed to see whether there is a signature there on a very personalized assessment of what's going on at the genetic level that would indicate that they are at a higher likelihood of recurrence and therefore need the chemotherapy, or whether they're at low risk and can be spared the cost and the side effects of what can be a pretty unpleasant experience. That personalized medicine intervention right now is saving our healthcare system this year about $100 million in terms of the women who won't end up needing chemotherapy who otherwise would go through it. So it's a pretty good example. Pharmacogenomics, another place where increasingly drugs that have different effects in different people based on their genetics, it's possible to assess that from drugs like abacavir for HIV, where it's now on the FDA label, essentially required to do a genetic test before you prescribe the drug in order to be sure that this is not an individual that's going to have a hypersensitivity reaction. Or even Plavix, which is, after all, one of the most highly prescribed drugs, and where there's now an FDA label saying physicians should be aware that about a third of the people given that drug will not benefit from it because of their genetics and should be offered some other alternative. So in those instances, I would say personalized medicine ain't the future, it's the present, it's here.
Well, those are all very exciting developments, and it sounds like everything comes back to global health. I do think we are out of time, but I want to thank you so much for talking with me today about these issues.
Okay, thanks, Angie.